My name is Robert Emmett, and my colleague David Rambo. Today I'm going to talk about plant disease, some concepts, causes, and then some of the diagnostic solutions that AgDia has for detection. So talk a little bit about concepts, some of the history, the impact of plant disease. Then we're going to talk about the three major pathogens, and then diagnostic testing options. So why do you test, and what products do we have available for you to use for testing? But before we jump into that, just a little bit of background on AgDia itself. AgDia is short for Agricultural Diagnostics. We are located in Elkhart, Indiana, and we're founded in 1981. So just a little bit of background on AgDia. So disease is defined as a progressive departure from normal form and or function caused by an infectious agent and manifested as symptoms. So keep symptoms in mind. We're going to talk about that a little bit later. It's probably been around as long as plants have existed and causes up to 20% crop losses around the world on an annual basis. So this crop loss impacts civilization through famine, population movement, disruption of economic markets, political and social unrest, and ecological change. So plant pathology is the study of plant disease and its effects on civilization. In Greek, the word pathos, where pathology comes from, means suffering or pity. And if you look left to right, these are all epidemics, either in the past or current, which have caused quite a bit of suffering for humans in one way or the other. So you have late blight, which is caused by Phytophthora, Panama disease caused by Fusarium, olive decline caused by Xylella fastidiosa, and this is chestnut blight. That's Cryphonectria parasitica. So plant disease as a concept is usually taught as a triangle. So this triangle, 3 points to the triangle, all 3 points have to be at one place at one time for disease to take place. So you have to have a susceptible host, a favorable environment, and a pathogen. So if any one of these is missing, no disease. When you get all of them, you get disease. Now, what I'm going to be doing is focusing primarily on the pathogen part of this triangle. And we're going to be talking about viruses, bacteria, and fungi. So one of the things that you want to keep in mind when you're diagnosing plant disease are two things: signs and symptoms. So a sign is an indication of disease from direct observation of a pathogen or its parts. Okay, so you have spores or the fungal body, which is a mycelia. And then symptoms would be indication of a disease by a physiological reaction from the host. So the sign is part of the pathogen. The symptom is the plant's response to the presence of the pathogen. Two very different things, but they go together. So jumping right into viruses, virus is Latin for virulentus, which means poisonous substance. The modern era of virology, plant virology and human virology, started with the discovery of tobacco mosaic virus. And it was first characterized by a microbiologist, this gentleman right here, named Martinus Beecherink in 1898. Viruses, a little bit different than the other plant pathogens. They're not considered living organisms because they do not reproduce and they contain no cells. They cannot replicate without a living host. Therefore, they are considered obligate intracellular parasites. Obligate because they cannot be outside of a host and be able to replicate. Intracellular because they live within the cell. They do their business within the cell. They're really just an infectious nucleic acid. With plants, plant pathogens, it's usually RNA, and they'll have a protein coat and a membrane. This is tobacco mosaic virus, or a representation of it. Viruses must be placed within the plant cell for infection to occur. And they can be spread in multiple different ways. So you have mechanical, which would be like pruning, tools, things of that nature. That's usually how they get spread locally. You can have insect vectors, which are very important. That can be either local or long distance spread. And then propagative materials. So propagative materials are typically how viruses get spread long distance. And propagative materials can be vegetative or seed. So typical symptoms of viruses, and one thing we need to understand is that viruses you can't see the signs of a virus. Remember, we talked about signs and symptoms. You need an electron microscope to see the signs of a virus. But the typical symptoms will include ring spots on foliage and fruit, mosaic patterns, tissue death, and then chlorosis and stunting. So there's not really any other way to know that a plant is infected with a virus without seeing the symptoms. Because like I said, you cannot see the signs without an electron microscope. So moving on to the next one here, it says viruses, but this is actually bacteria. This is bacteria. So bacteria are prokaryotes. So they are single-celled organisms. Unlike viruses, they are an organism, and most have cell walls. Now, the first bacterial pathogen to be characterized was Erwinia amylavera, which causes fire blight on apple and pear. And it was first characterized by this gentleman here, Thomas Burrell, in 1878 at the University of Illinois. So once again, these are bacteria, not viruses on this one. Okay, so once again, viruses up there, it should be bacteria. They multiply by asexual fission, so they basically just divide, and they infect the plants through stomata, natural openings, wounds, and vectors. These are bacteria growing into a stomate on a plant leaf. Bacteria are typically spread wind and rain. So hurricanes, typhoons are notorious for spreading bacteria. Human activity, insect vectors, and infected plant parts. So bacteria will produce enzymes, toxins, and a substance called EPS, or extracellular polysaccharides. Some people call it ooze, but it's this here. It's dripping out of the plants or in the vascular system of the plant. This is a sign of a bacterial infection. So if you have bacterial ooze like this, it's going to be a bacterial infection. Typical symptoms are going to include wilts, leaf and fruit spots, soft rots, and galls. So the next group I'm going to talk about, probably the biggest, it's definitely the biggest group, would be the fungi and the oomycetes. So these are multicellular eukaryotic organisms, so they have a true nucleus. And fungi make up about 85% of the plant diseases that infect plants. So viruses and bacteria, and then basically fungi make up just about everything, well, 85%. So the beginning of the study of fungi was really a gentleman named Anton de Berry. He's considered the father of plant pathology. He studied both wheat rust and late blight. Wheat rust is a fungi, late blight is an oomycete. And he published his work on the Irish potato famine, which was caused by late blight. And he did that in 1861 through 1863. So fungi are capable of sexual and asexual reproduction. So they can do both. The body of the fungi is known as the mycelium. They produce spores as infectious propagules. So the mycelia or the spores can infect the plant. But what happens is these produce spores, the mycelia produce spores, the spores land on a plant, they germinate, form more mycelia, and the cycle continues. Out of the pathogens we're talking about, fungi are the only ones that can actively penetrate plant tissue to infect the plant. So the way that these guys are spread, fungi and oomycetes, once again, wind and rain, human activity, infested soil, infested irrigation water, infected plant materials, and a lot of fungi can actually release spores on their own. It's a forced release. So they blow their spores into the air on their own. Now, typical symptoms of fungal and oomycete infections, vascular wilt, leaf spots, stem cankers, root rots, and wood decay. So now that we've talked about the pathogens a little bit and everybody has a little bit of background on these, I think the next thing we jump into is discussing why you want to test. So why do you want to use AgDia's products? Why do you want to test your plants to make sure that they're not infected with a virus, a bacteria, or a fungi? Well, that's because prevention by exclusion is the best management practice. And you have to identify the problem before you can develop a solution. So the diagnostic products I'm going to talk about are called serological tests. So they are Immunostrip and ELISA are the products that we sell. So these are based on immunochemistry, and it's immunochemistry because it's what happens in the immune system. They are sensitive and they are specific at times, and they are based on a reaction or a binding that takes place between an antibody and an antigen. So this would be on the pathogen. This would be the virus, on the bacteria, on the fungi, and this antibody binds to that. So the reaction between the antibody and the antigen causes lines on the Immunostrip or color change in the ELISA plate. So focusing on the Immunostrip, it includes all the necessary components. So everything you need comes with that. It's user-friendly, can be used by lab and field personnel. No experience is required. It will give you a positive or negative result. So no guessing, it just either positive or negative, and you typically get results within 30 minutes. And this is a list of the immunostrips that we have available for a lot of the pathogens that you had listed that were of importance to your to the rest of the people involved here. So ELISA stands for enzyme-linked immunosorbent assay. It's really the most common serological test. It's been used for over 50 years, and it's used by labs around the world, including a lot of the big seed producers. It's the most economical method, and you can test multiple samples at one time. So ELISA is a very popular method for testing. And once again, these are the pathogens that we have available in ELISA for ELISA format. So I'm not going to go through all of them, but hopefully that your guests have the slides so they can look at those. And that is the end of my presentation. So if you have any questions,